Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The intellectual landscape of biology in the mid-20th century was dominated by a central, profound enigma: the genetic coding problem. Following the elucidation of the deoxyribonucleic acid double helix, it became axiomatic that the linear sequence of 4 nucleotide bases must somehow specify the linear sequence of 20 amino acids that constitute proteins. This informational transfer represented the very essence of life's continuity and function, yet the specific language, the precise rules of translation, remained a realm of fertile theory and frustrating experimental impasse. Into this environment of intense speculation and methodological searching appeared the 1961 paper by Marshall Warren Nirenberg and Johann Heinrich Matthaei, published in the Proceedings of the National Academy of Sciences, titled The Dependence of Cell-Free Protein Synthesis in E. coli upon Naturally Occurring or Synthetic Polyribonucleotides. This work represented a monumental breakthrough, its significance derived from an experimental design of sublime simplicity and profound consequence, a design that bypassed the complexities of the living cell to pose a direct biochemical question to the machinery of life. This essay provides a systematic, section-by-section evaluation of the internal claims, methodological rigor, and interpretive logic presented within this seminal publication, analyzing how the authors constructed their argument from the ground up, moving from the validation of a novel system to the triumphant deciphering of the first word in the genetic lexicon. Section 1: The conceptual and methodological framework. The paper's initial sections lay the groundwork by articulating the development of a reliable cell-free system derived from Escherichia coli, a system capable of protein synthesis. The core claim here is the successful creation of a tractable experimental environment where the components of translation could be manipulated and their effects measured directly. The authors detail the preparation of what they term the S30 fraction, a supernatant obtained after centrifuging disrupted E. coli cells at 30,000 times the force of gravity. This fraction contained ribosomes, transfer RNAs, aminosylt RNA synthetases, and other soluble factors essential for polypeptide synthesis. The brilliance of this methodological claim rests on what the system includes and, more importantly, what it can be made to exclude. The authors report the deliberate addition of deoxyribonuclease, or DNase, to the preparations. The purpose of this treatment was to degrade the cell's native genomic DNA, thereby uncoupling protein synthesis from the primary genetic template. This single step was conceptually critical, creating a system that was inert until programmed with an external source of information. The authors claim this system, supplemented with an ATP-generating source and a full complement of amino acids, becomes dependent upon the addition of messenger ribonucleic acid, or mRNA. The paper's initial argument is therefore about control and potential. It posits that a complex biological process has been reduced to a stable, manipulable and information-starved biochemical reaction in a test tube, waiting a script. The evaluation of this claim is positive. The thoroughness of the preparation protocol, including the pre-incubation step to reduce endogenous synthesis, demonstrates a deep understanding of the necessary controls to isolate the phenomenon of interest. They constructed a biological stage and cleared it of its resident actors, preparing for a performance directed by a new script of their own choosing. Section 2: The foundational experiment demonstrating RNA dependence. Following the description of their system, Nirenberg and Matthaei present data to validate its central premise. The key claim evaluated in this section is that protein synthesis in their S30 extract is, in fact, directed by ribonucleic acid. This is a foundational assertion upon which all subsequent findings depend. The evidence is presented in the paper's first data table, which quantifies the incorporation of carbon-14 labeled valine into protein under various conditions. The control experiments are models of logical scientific inquiry. The baseline incorporation of the labeled amino acid is established. The addition of deoxyribonuclease, the enzyme that degrades DNA, produces a negligible change in this baseline activity. This result supports the contention that the system functions independently of the bacterial chromosome, a crucial element of the experimental design. The subsequent experiment involved the addition of ribonuclease, or RNase, an enzyme that specifically degrades RNA. The data show that this treatment causes a dramatic reduction in labeled valine incorporation, diminishing the synthetic activity by over 90%. The logical force of these paired experiments is formidable. By demonstrating the system's indifference to the absence of DNA and its profound sensitivity to the absence of RNA, the authors provide compelling quantitative evidence for their claim. The system is RNA-dependent. This section successfully transforms the methodological assertion from the previous section into a verified experimental reality. The data are clean, the effect is large, and the conclusion is direct. They have validated their tool, proving its suitability for the primary investigation into the nature of the informational molecule. Section 3. The pivotal discovery, polyuridylic acid and phenylalanine. This section of the paper presents its most celebrated and transformative finding. The central claim is that a synthetic ribonucleic acid polymer of a single, repeating base can direct the synthesis of a specific polypeptide. Having established an RNA-dependent system, the authors made the audacious leap of introducing a synthetic messenger, an artificial script. They chose polyuridylic acid, a long-chain molecule consisting solely of uracil-containing ribonucleotides. The experimental design was beautifully straightforward. 20 parallel reactions were prepared. Each contained the S30 cell-free system, the ATP-generating components, and polyuridylic acid. Each of the 20 tubes, however, contained a different amino acid labeled with radioactive carbon-14, while the other 19 amino acids were present in a non-radioactive form. This allowed for the specific tracking of any single amino acid's incorporation into a newly synthesized protein. The results, presented in the paper's iconic first figure, were stunningly clear. In the tube containing labeled phenylalanine, the incorporation of radioactivity was immense. The authors report that the addition of polyuridylic acid stimulated the incorporation of phenylalanine approximately 1,000-fold over the baseline. The count per minute, a measure of radioactivity, surged from a background level of fewer than 50 to nearly 40,000. In stark contrast, the other 19 tubes containing the other labeled amino acids showed little to no stimulation. The sheer magnitude of this effect lends the claim its profound power. This was a signal of exceptional strength and clarity, rising dramatically above the biochemical noise of the system. The finding was unambiguous. The synthetic message, uracil was being translated by the cellular machinery into the polypeptide phenylalanine. It was the first time a direct, causal link between a specific nucleic acid sequence and a specific amino acid had ever been established experimentally. Section 4. Establishing specificity and the nature of the product. A scientific claim of this magnitude requires rigorous corroboration. This section of the paper addresses two vital ancillary claims. First, that the stimulation effect is highly specific to the phenylalanine polyuridylic acid pairing, and second, that the radioactive product formed is indeed a polypeptide composed of phenylalanine. The evidence for the first claim comes from a table summarizing the results for all 20 amino acids. The data confirm the visual impression from the earlier figure, showing massive stimulation for phenylalanine and only trace or background levels for every other amino acid. This exhaustive survey rules out the possibility of a general, non-specific stimulation of protein synthesis and powerfully reinforces the concept of a specific code. The system was reading the polyuridylic acid and selectively incorporating the one correct amino acid to address the second claim regarding the product's identity. The authors performed further biochemical analysis. They demonstrated that the radioactive product was precipitable with trichloroacetic acid, a classic characteristic of high molecular weight polymers like proteins, distinguishing it from free amino acids. Furthermore, they subjected the product to acid hydrolysis, a procedure that breaks polypeptides down into their constituent amino acids. Subsequent paper chromatography of the hydrolyzed material revealed that the radioactivity commigrated precisely with a known standard of phenylalanine. This chemical verification was the final, critical piece of the puzzle. It proved that the system had manufactured a genuine polypeptide, and that this polypeptide was, as predicted, polyphenylalanine. The internal logic is impeccable. They observed the phenomenon dramatic incorporation, demonstrated its specificity only phenylalanine, and then confirmed the chemical identity of the resulting product. The argument was sealed. Section 5 expanding the lexicon, the introduction of copolymers, the deciphering of one codon, while historic, was a single data point. The subsequent section of the paper evaluates the claim that this cell-free system could serve as a general tool for deciphering the entire genetic code. To test this, Nirenberg and Matthaei moved beyond homopolymers and began to use synthetic copolymers, which are RNA strands containing a random mixture of two or more different bases. The paper specifically details results using a polyuridylic adenoic acid copolymer and a polyuridylic etidylic acid copolymer. The introduction of these more complex messengers led to a corresponding complexity in the results. For example, the polyuridylic etidylic acid template stimulated the incorporation of not only phenylalanine but also serine and proline. The logic was that the presence of cytosine in the messenger RNA, in addition to uracil, created new codons that specified these other amino acids, while the random nature of the copolymers prevented the authors from determining the exact sequence of the codons. For instance, they could not distinguish between a UUC, OCU, or a CUU codon for serine, and results were profoundly indicative. They demonstrated that the system responded predictably to changes in the composition of the messenger RNA. The claim being evaluated here is one of generalizability and utility. The data strongly support this claim, showing that the experimental approach was extensible. It transformed the problem from a singular discovery into a systematic program of research. The results with copolymers served as a powerful proof of concept that the same ASIC technique, with further refinement, could be used to assign amino acids to a wide variety of base combinations, thereby filling out the entire genetic dictionary. This section was less about a single, clean answer and more about demonstrating the immense future potential of their methodology. Section 6, a cautious and prophetic conclusion, the final section of the Nirenberg and Matthaei paper presents their interpretation of the data and a vision for the future. The claims here are interpretive and prospective. The authors, with a degree of scientific circumspection that enhances the paper's authority, conclude that their results suggest that polyuridylic acid contains the information for the synthesis of a protein having many of the characteristics of polyphenylalanine. The careful phrasing, using suggests and many of the characteristics, reflects an appropriate humility in the face of a revolutionary finding. They explicitly discuss the coding problem, noting that if the code were a triplet, then the sequence of 3 uridylic acid residues would be the codon for phenylalanine. However, they are careful to state that their data alone could not resolve the exact size of the codon. This intellectual honesty, a refusal to overstate the conclusions, is a hallmark of a classic scientific work. The most important claim in this section is implicit that the coding problem, once a matter of abstract theory and genetic inference, was now solvable by direct biochemical experiment. They had established the paradigm. The conclusion was a roadmap for their own laboratory and for the entire field of molecular biology. The ensuing years, which saw Nirenberg, alongside teams led by Severo Ochoa and Hargobind Gorana, rapidly fill in the details of the code using refinements of this very technique, are a testament to the accuracy of this prophetic claim. The paper concludes by providing the key to the Rosetta Stone of life and, just as importantly, by describing the very method of its use. In conclusion, the 1961 paper by Nirenberg and Matthaei stands as a masterpiece of experimental biology, its power deriving from a cascade of meticulously validated claims. The authors first demonstrated the successful construction of a controllable cell-free protein synthesis system, liberating the translational machinery from the confines of the living cell. They then rigorously proved this system's dependence on an RNA template, setting the stage for their central experiment, the subsequent demonstration that a simple, synthetic polyuridylic acid messenger could direct the massive and specific synthesis of polyphenylalanine was an electrifying result, providing the first direct solution to any part of the genetic code. Their work went further, establishing the chemical identity of the product and showing through the use of copolymers that their method was a generalizable tool for deciphering the entire code. Each step of their argument was supported by clear, quantitative data and logical controls, building an edifice of certainty around a conclusion of profound significance. The work's enduring legacy is rooted in this methodological elegance and evidentiary force. It transformed the abstract puzzle of the genetic code into a tangible, biochemical problem and, in doing so, provided both the first answer and the means to obtain all the others, launching a new and definitive chapter in the understanding of life's fundamental processes.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.